Mendelian genetics. So this is the first of two lectures you should be watching over break. This covers basic genetics and the laws of inheritance. When you come back from spring break, we'll talk about the advanced genetics. So what are we discussing in this lecture? We're going to be talking about who is the father of genetics, what are the definition of many of the genetics vocab you'll need for the next few weeks, how genes are combined to create new individuals, and we'll wrap up discussing Mendel's laws which govern the inheritance. So Gregor Johann Mendel. He was an Austrian monk. He studied pea plants and how traits were passed along from generation to generation. He was the one that developed the laws of how genes are passed along and how traits are passed from offspring to offspring. He was born in the 1800s, but he was not really recognized for his work until the following century. So between 1856 and 1863, Mendel cultivated 28,000 pea plants. What he found out was that the offspring of the pea plants had some of the traits of the parents. So he discovered that genes or traits are actually passed along from parent to offspring. Because of this discovery and the laws he came up with, he is known as the father of genetics. So Mendel knew that traits were passed along from person to person and from parent to offspring, but he did not know exactly what they were or what they were called, so he knew that they were some kind of particle. He did not know that particles were actually genes because they didn't know DNA existed at this point in time. So you also need to remember that on a DNA strand, there are little bits of segments that code for a protein. That is what we call a gene. So a DNA has thousands of genes encoded on it. Alright, so here's some genetics terminology. First thing is known as a trait. So a trait is any characteristic that can be passed from parent to offspring. Hair color, eye color, skin color, all of those are traits. Heredity literally means the passing of traits from parent to offspring. And genetics is the study of heredity. So genetics is the study of how traits are passed along from generation to generation. So there'll be two types of crosses or breedings that we'll be talking about. The first is known as a monohybrid cross, which involves a single trait, such as flower color. So you breed one red flower with one white flower and see what comes out of it, what the kids look like. Another is known as a dihybrid cross, which is when you have two different traits that you're crossing. So example could be a tall red flower with a short white flower, and then seeing what the offspring look like. So something you probably remember from middle school are these Punnett Squares, which are used to determine the likelihood of a certain offspring coming about from the mating of two parents. So we'll be using these a lot over the next few weeks, and this is the main topic of the next video, is showing you how to set up a Punnett Square, how to use them, what they mean. So it's something that you're definitely going to have to get comfortable with. So we have a few more vocab words on this slide. A few slides ago we talked about how genes are the physical object that get passed from parent to offspring. Well, clearly you don't look exactly like your parents, and you don't look exactly like the person you're sitting next to in class. So for every gene, there are multiple forms of that gene. So an example of that is hair color. Everyone has a gene that controls their hair color. But the gene that you have could be slightly different than the gene of the person next to you. We call those differences, or those forms of a gene, an allele. So an allele is just a different form of a gene. Brown hair versus blonde hair. Blue eyes versus brown eyes. The same gene, just slightly different forms of it. Usually there's only two forms of a gene. One of them is known as dominant. The other one is known as recessive. So a dominant allele is the stronger of the two alleles and is always expressed. Because it is dominant, when we talk about genotypes, it is always represented by a capital letter. The other one, the recessive, is weaker, so it does not show up when the dominant gene is present. So we use a lowercase letter to represent that. So genotype versus phenotype. So a genotype is the combination of genes that you have for a trait. So example, it could be big R, which means you have both dominant alleles. It could be big R, which means you have one dominant, one recessive. Or it could be little r. Your genotype will always be 2 letters because you get one from your mom, one from your dad. A phenotype is what you physically look at, or you physically look like. So the genotype causes the phenotype. So the genotype might be big B. The phenotype might be brown hair. So a genotype determines your phenotype. Alright, so let's show an example of this. So there's 2 different alleles for flower color. Big R represents the red flower. Little r represents the yellow flower. It is traditional to use the letter of the dominant allele. So that's why the yellow flower is represented by a little r. Because red being dominant, we use the big R. The recessive version, the yellow version, we use the little r. Alright, so you can see on the bottom there's 3 different possible combinations of genotypes. You can have dominant, big R. You can have dominant recessive, big R, little r. And you can have complete recessive, little r. If you look at the phenotypes, however, the dominant allele is red. So when you have dominant, your phenotype is red. Once again, the red is dominant. So when you have big R, little r, the big R masks the little r. So the flower still appears red. And then you have little r, which is recessive, which you actually get to see the yellow color. So if a person has one copy of the dominant allele, it is the one that is usually shown. So there are 2 different types of genotypes. The first is known as a homozygous genotype because homo means the same. So homozygous means that you have the same 2 alleles. In other words, you can be homozygous dominant, in which case you have big R. You can be homozygous recessive, in which case you have little r. This is known as being purebred because you are exactly the same. You have the same copy from mom and dad. When you have a mixture, we call that heterozygous because a hetero means different. So a heterozygote has one copy of the dominant, one copy of the recessive, or sometimes we refer to them as a hybrid because they have one of each. Alright, so we're going to talk a little bit about Mendel's pea plant experiments and how he discovered his laws and what he came up with. So why did he use pea plants? Peas can be grown in a small area, and Gregor Mendel was a monk, so he only had a very small little vegetable garden to do his experiments in. 2, peas produce a ton of offspring, so you don't have to wait a very long time for them to regrow and regenerate and make a new generation. Imagine if you used humans, you have to wait good 15, 16 years before being able to breed them again. Also, you can produce pure plants when you just let them self-pollinate, so you don't let them mix pollen with different plants, you just leave them alone. And you can artificially cross-pollinate them, so you can physically move pollen from plant to plant to get the crosses that you want. So we already know a little bit about flowering plants. We know that pollen is the sperm of a plant and it's produced by the stamen. We know that the ovary has the eggs, which is part of the pistil of the plant. So for a plant to reproduce, the pollen has to get from the stamen to the pistil. Alright, so how did he actually conduct his experiments? So what Mendel did was he hand pollinated flowers using a paintbrush. So he literally went from flower to flower, collecting pollen in the paintbrush, moving it to the next one, and dropping it on the pistil. He then snipped the stamens of the ones that he collected pollen from so they could no longer actually cross-pollinate or let their pollen flow. The ones that he did pollinate, the ones that he put the pollen in the pistil, he then covered with a cloth bag so that none of the pollen could escape and no more pollen could get in accidentally. So when we talk about generations or genetics, there's a certain order that we denote the generations. So we call the parent generation the P1 generation. So that is the generation you're actually testing and crossing yourself, you're breeding yourself. The result of the P1 generation is known as the F1 generation, which is the first generation of offspring in an experiment. So if you are the parents, you have a child, that is the F1 generation. The next generation, when you breed two F1s together, is called the F2 generation. And then you just keep on going and going, F3, F4. But we don't go past the F2 generation in our class. Alright, so let's try to follow the generations here. So we have two pure plants originally, big T, little s. We breed them together, the P1 generation. That results in an F1 generation of all hybrids, big T, little s. We then cross two of those hybrids together, so we inbreed two of the hybrids that were just made, a brother and a sister. That results in 3 tall plants and 1 short plant. And if we actually look at the genotypes, we would see that's big T, homozygous dominant, big T, little s, heterozygous, and little s, heterozygous recessive. So you see from the parent P1 to the F1 to the F2 generation. So now to wrap it up with a little discussion of Mendel's 3 laws. So his first law is known as the law of dominance. So the law of dominance says, we already mentioned this, that if you breed 2 plants together and the result is a hybrid or a heterozygote, where you have 1 dominant allele and 1 recessive allele, the dominant is the trait that is shown. So you can see an example in the Punnett square to the right. You have a purebred green, which is big G on the left. You have a purebred yellow bean, which is little g. You breed them together. The genotype of all of the offspring are big G, so they're all heterozygotes. However, the phenotype of all of them, what they physically look like, they're all green, because the green is dominant, so it overpowers the yellow. So you don't see the yellow at all. All you see is a perfectly green bean. So another one of Mendel's laws is known as the law of segregation. So during meiosis, we talked about that's how the process of making sex cells of your gametes. So we all have 2 copies of a gene. So we talked about how we have 23 pairs of homologous chromosomes. 1 from mom, 1 from dad. So when the gametes are formed, those homologous chromosomes are split. So when you make a sex cell, a gamete, you either pass along the gene from your dad, or you pass along the gene from your mom. So we call this the law of segregation, where your 2 genes are segregated into different gametes. So you can see in this case of this dog, he is homozygous recessive, little b. When he makes sex cells, the little b's will separate. One will go to one sex cell, the other will go to the other sex cell. So that's the law of segregation. You are segregating your alleles into your sperm cell or egg cell. Alright, so let's do an example here. So we have this little bird, which is heterozygous. So it has 1 dominant, 1 recessive. Due to the law of segregation, this capital Y will go to one of them, and this lowercase y will go to the other one. So you can see how they are segregated. So either you're going to pass along this one, another example, or you're going to pass along that one there. It's homozygous recessive to little r's. So I'll give you a few seconds. How will they segregate into the egg or sperm cells? So hopefully you see that they split. So each sperm cell or egg cell gets a little R in it. So therefore, this flower can only pass along genes for the white coloring. Alright, so let's do a little more complex example. So now let's actually breed two flowers together. So we have a red flower and a white flower. So the red flower is a heterozygote, so it has a dominant and a recessive, big R, little r. The female white flower is homozygous recessive, so it is little r, little r. So due to the law of segregation, the big R and little r will split. And for the white female flower, the little r and little r will split. And now we'll see what the possible combinations happen when we actually breed these two together. Alright, so sperm 1 was big R from the previous slide, so we can fill those in with big R's. Sperm 2 was little r from our previous slide, so we fill those in with little r's. Egg 1, or all the eggs, egg 1 and egg 2, all only could pass along little r's, so we can fill all those out. So when we breed sperm 1 with egg 1, we literally just breed those two together. We see the genotype is big R, little r. Big R from there, little r from the other one. For the next one, we see it's big R, little r. This one we see it's little r, little r. And then finally we have little r, little r. Now for the phenotype. So the big R represented red, which was dominant. The little r represented white, which was recessive. So anytime we see the big R, we know that the red is going to be shown. So for the first two flowers, they're going to appear red to your eyes. Whoops. Red. Red. Oh, terrible writing. For the other two, they're homozygous recessive, so they do not have a dominant gene. Therefore, they're going to be white, which I'm not going to write out. I'm just going to put a W for white. So you can see the cross between these two flowers, the red and the white flower, 50% of their offspring would be red. 50% of their offspring would be white. So then that's due to the law of segregation. You can see how they segregated. The allele split in the parent when they made their gametes. So the final Mendel's law is known as the law of independent assortment, which means that the alleles for different traits are distributed independently of each other. In other words, if you are blonde hair, blue-eyed, just because you passed along a blonde gene does not mean you're guaranteed of passing along a blue gene. So they are completely separate. So your height genes are independently sorted from your eye color genes, from your hair genes, from every other gene. So pretty much nothing in your body is actually linked together. Now when we get back from spring break, we'll talk about gene linkage, but most of your traits are independently assorted, which we'll discuss when we do dihybrid crosses when we get back. So this is it. That was the end of this lecture covering basic Mendelian genetics. Hopefully you filled in your notes as we were going along like you're supposed to. So we talked about who Mendel was, how he was the father of genetics. We discussed many of the vocab words about genetics. So we have homozygous, heterozygous, dominant, recessive, gene versus allele, law of independent assortment, law of segregation, law of dominance, what a Punnett square is. So there's a lot of information in this section. This also is chapter 6 in your textbook. So if you want to read through that to make sure you got everything. And then you can finally, this last little comic here wraps everything up. Remember that there's another lecture you should watch after this one called Punnett Squares, which I'll go through a practice of how to do a Punnett square, and then you have the worksheet that you're given to fill in and practice doing Punnett squares.